all these talks. So the next talk is from a veteran expert in the field of prion disease, Joaquín Castilla. He'll be talking about a very important topic, especially when we go into evaluation of prion therapeutics and looking at different models to study that. And his talk is entitled Development and Characterization of the Fastest Animal Model Able to Propagate GSS Prions Faithfully.
Thank you very much. First of all, I would like to thank to the foundation and to all families to make possible this study. As I mentioned, as Paul B mentioned, we are gonna show you the development and characterization of a new animal model. If you want to test new therapeutical approaches, you need a very good animal model. This is absolutely necessary. The preclinical studies are absolutely necessary to go to the human. For that reason, our project aims to develop and characterize this model, specifically for GSS prions. Briefly, I'm gonna just give some data that this particular prion disease show is an autosomal dominant hereditary disease. Is ultra rare disease, approximately affect 60 families worldwide, and the age of onset can vary between 30 to 60 years old, and the duration is longer than usual comparing the other prion diseases from 3 to 10 years. The clinical manifestations include also several ataxia, pyramidal signs, and dementia, and is, as usual, very heterogenic. Several mutations are involved in this disease. These are the least, and in this particular study, we are going to focus on these two mutations are most prevalent, P102L and A117V. For decades, these GSS prion diseases were considered a non-infectious and non-transmissible prion disease. And this project is going to show a completely different result, because if you want a good animal model, you need to have these kind of characteristics. The model should exhibit pathological hallmarks of the neurodegenerative disease, display clinical signs that resemble the symptoms observed in humans, but should also exhibit this progressive course of the disease, mirroring the temporal progression of pathology. But also, it should help to evaluate the efficacy of potential therapies and study underlying mechanisms of action, and should be also accessible, reproducible, and manageable in terms of breeding, housing, and experimental procedures. The models that fit with all these characteristics are going to be very valuable for understanding disease mechanisms, testing potential therapies, and advances our knowledge of neurodegenerative disorders. In this project, we have concluded 6 different studies. Comparative study of different animal models I'm going to show you later. The specific infectivity, that is basically how infectious is the inoculum, the prion disease in this particular model. A study of assess the impact of the PRP quantity that I'm going to show you later, the importance of that. Kinetics for the study of biomarkers. Kinetics to understand the temporal progression of the disease. And a proof of concept, how this model can be used for the real therapy or therapeutical approach. I'm going to focus on 2 different isolates or different GSS samples, A117V and P102L. And these are the models that we use as control, the wild-type mouse, and the mouse or the transgenic mouse expressing human PRP, the humanized models. In the first case, we have just 1x expression of the protein, and in the humanized model, we have more than 6x of expression. And then are these three models. The first one express the Van Vogt PRP, that is a rodent protein, in just around 1x. The other one, 3x of the mouse PRP, and the third model is going to express 4x of the same rodent PRP. The first study was the comparative study of these different models. When you use wild-type mouse and inoculate with these two isolate of GSS, two different type of GSS, you don't have disease. When you use the same same isolates, but in the humanized, even if they are humanized model, you don't have disease. That's the reason why this GSS was considered non-infectious. When we use the new models, the Van Vogt models, 1x, we are starting showing a clear disease in a very much shorter incubation times. But when we use these particular isolates, 3 different isolates, we obtain a very, very short incubation time, close to 70 days. When we use the overexpressor using mouse PRP, this was reduced less than 60 days. And when we use a Van Vogt overexpressing 4x, we have a reduction in both cases. In particular case, this isolate, less than 40 days. That this is an amazing short. In these 3 models, we have a PRP containing a specific change, isoleucine in the position 108 or 9, depending if it's mouse or if it's Van Vogt. When we did this intraperitoneally instead of intracerebrally, that is very, very useful for any therapeutical approach, we observe also, this is ongoing, but with this particular isolate, we could observe less than 90 days of incubation after intraperitoneal inoculation, that is always longer. When we use the other models, we have less than 60 days or 50 or 55 days after intraperitoneal inoculation. This is indicating that are going to be very useful for any kind of therapeutical approach against GSS. When we did the second passages, serial passages, these are the first passages in 2 different models, we observed something that we didn't expect. Instead of a reduction of the incubation time because we were using something that was adapted from the previous passages, we observed something that, to be honest, we cannot explain yet, but is happening in both cases. If we want to work with a very good model, we have to reproduce the disease, and this is what happened. These are different slides of the same brain, and the dots are indicating the accumulation in brown or in white, the spongiosis. So basically, these diseases are showing these depositions in the right place, in this particular case in hippocampus, or in this place also in hippocampus, striatum, and occipital cortex, and of course in cerebellum, as is common in these diseases. Also, the biochemical studies indicating the typical future, a very small band indicating for these atypical GSS cases. In the original one, human, but also after the TG mouse and the TG Vogt models. So this is also correlated very well what we're expecting. The specific infectivity is another study that we did in this project just to show how infectious is really this isolate in this model. We use different dilution with this GSS from 10 to minus 1 to 10 to minus 8. And we show this very nice correlation, observing that we have enormous amount of infectivity that is almost 10 to 11 ID50 per gram. So this is very useful also for therapeutical approaches because we can use different dilution and still have short incubation time. So the next study was to demonstrate that the impact of reducing PRP was really a very good therapeutical approach. And what we did is just to compare the low dilutions from these isolates, and even showing also short incubation time, 66 days, and using now the same model, but reducing the half of the protein, in order to demonstrate that even this model can be infected, but the amount of with a double time, indicating that the reducing of 50% of the PRP has an enormous impact. When we did with another dilution that still was reasonable in time, 80 days, we have also a double in the incubation time. So this is indicating that these nice studies from Sonia and Eric are very promising. And in my opinion, this model should be used also if you want to demonstrate the effect of the reducing this PRP in this particular prion, GSS prion. Let's talk about kinetics for this study and using blood-based biomarkers, because we don't want to, or we don't need to go to the end of the disease to have data. So, but how far we can go? So we are going to use in this case, well, we use NFL. It's a neurophenomenon light chains. It's a very good marker. We inoculate the animals intracerebrally, and we extract blood from the same animals, different groups, at different timing. And the same with intraperitoneally. More or less each 2 weeks, the animals were bled. And what we observed using a very sensitive technology, and this is a collaboration with a group of Marcus Otto, we observed that the controls were negative as expected, and the NFL was observed in around 20 days before that. For this particular incubation time is a lot of time before the disease. The same was for the intraperitoneal inoculation, observing negative controls, and that the NFL was able to visualize around 20 to even 25 days before. That this is very useful for any kind of therapeutical approach. If you can go even to pre-symptomatic time to demonstrate that the therapy is working. And what about where the prion is starting in the encephalon? So we started doing inoculation, intraperitoneal inoculation, and extracted after 2 weeks, each 1 week, groups of around 6 animals, and we evaluate the level of PRP rest and spongiosis observed in these animals. So in this, the first cases appear here, and what we observe basically is a unilateral spongiform lesion, or in other cases also bilateral, but starting in the hippocampus. So this is very useful also if we want to, and this is happening also in these pre-symptomatic cases. So we know that an intervention, for example, intraventrically, is going to be very useful because it's very close to the area that is where the progression of the disease is starting. And finally, the last study is to demonstrate that this model can be used for standard antiprion therapies. In this particular example that we did the last year, we use an intraperitoneal inoculation of the prion, and this is the animal model that we are discussing. And in this particular case, this model that express or is producing continuously an antiprion compound based on porphyrin. When the animals were inoculated with prions, in the control cases, we have 100% of the animals dying in less than 90 days. But in this model that continuously produced this porphyrin, we have enormous delay with more than 200. So this is the proof that for this particular GSS prion, we can use this model to demonstrate that this type of porphyrins are very, very active. Just summarizing, we have developing the fastest animal model to date for a human prion disease. Very useful by this to evaluate different inoculation routes, and is demonstrated that it are very suitable for testing treatments. We have examined the brain lesions that develop in these diseases, and we know where the disease is starting. And we have also analyzed the arousal of a biomarker in blood, the level of which increased weeks before the onset of neurological clinical signs. And we have demonstrated that can be very useful, this model, to assess the efficacy of, in this particular case, two potential treatments. One treatment aims to reduce PRPC levels, and the other one based on a molecule with antiprion activity. I want again to thank to the families that have made possible this study, and also to my group and my colleagues that have been very useful and necessary to this study. Thank you very much.